In this video, we will discuss the paper 5 of A-Level Biology 9700. In Cambridge International Biology 9700, paper 5 is called Planning, Analysis and Evaluation. So it has higher order practical skills, not factual recall. It has three main components. So this paper, it is of 30 marks. 30 marks, and the time is 1 hour 15 minutes to solve this paper of 30 marks. So this paper consists of three main components which you must know how to solve this paper. So first component which we are going to discuss first is the planning. And planning is about designing an experiment or investigation. So for this purpose, in this question, which can be of question 1, you must design a complete workable experiment to investigate a given problem. That is, one problem will be given in the paper, and according to that, you have to solve that paper, that you will plan an investigation, and this planning of investigation can be of 8 marks. So you are expected to, when you are going to solve that planning, you are expected to, first of all, to state a clear hypothesis. Clear hypothesis, what is hypothesis? Untested statement. So before designing an experiment, or you have to write or state a clear hypothesis. And hypothesis should be includes a biological relationship, not just a prediction. That is what you are going to plan. So it have any relationship between. So then after this, after planning a hypothesis, after stating a hypothesis, then you have to identify the independent, dependent, and controlled variables. Independent variables and dependent variables and controlled variables, which you have constant variables, that is during the, which remain constant during the experiment, and independent, according to which the, you can say, the dependent variable will be changed. Then after this, you come to describe a valid method. You have to write a method with the help of which you are going to um, analyze a data. So you have to fix a data. You have to go into that. So how to vary the independent variable. So how to vary the independent variable? You must know that how to measure the dependent variable. You have to include all these things into your answer. So how to control variables? You must know all these things. You must include all these things into your answer, which you are going to write for this planning. Then how to control variables? Which variables you must know, which should be controlled? They are volume, concentration, time, temperature, etc. So these are the major which should be controlled. After this, the next, what should be present, that is to, you have to describe a suitable apparatus. You have to describe a suitable apparatus, how to fix that apparatus, how to set that apparatus for that investigation. All these things are needed to write in your answer script. Then you have to explain the risk factors. Risk factors or safety precautions all should be written in the answer. Predict results or explain how results relate to the hypothesis. So then this section tests the ability to think like a scientist. That is, you have to think that you are a scientist, that you have to plan that investigation, how to plan that investigation, how to control the variables, how to know how to control the variables and how to predict the hypothesis and how to manage the independent variable and how to set the apparatus, and the use of different chemicals for that investigations. You must know all these things. Means all they need, all this is called as a planning, and planning of an investigation or experiment. So then after planning, the second part of this paper is you will be given raw experimental results or a graph to interpret. You may need to calculate. So there will be some calculations. There will be given data, or with the help of that data, you have to draw a graph. You have to interpret a graph. So you may need to calculate what? You may need to calculate what? Means, rate, percentages, ratios, standard deviation, or uncertainty, or identify anomalies. So all these things you need to calculate. Any one of them or any two of them. It depends on the paper. So then after knowing all these things, all this, you know how to draw or complete a graph. Graph has been given. You must know on x-axis there should be independent variable, on y-axis there should be dependent variables. Then you will be able to draw a graph. Then comment or patterns. The next question which can be, that is, you have to describe the trends or patterns which you have drawn on the graph. Then identify the relationship between the variables. All variables which have been given in the paper, you must know the relationship between those variables. Then you can see that in this section, check your ability to handle practical data. In the first component, you must think like a scientist, and in the second, this section checks your ability to handle practical data, how to handle the practical data which has been given in the paper. Then we will move towards the third part, which is on evaluation, in which you are going to assess an experiment. So for this purpose, you evaluate the experiment described in the question. You need to discuss, you must know what is the source of error during that investigation which you have designed, or in which they have given to process or to draw a graph. During that investigation, what can be the source of error? You must know. You must think like a scientist, that is, what can be a source of error during that investigation. So it can be random or systematic. Then the limitations in the method improvements. How to improve the method? They can ask from you that how to improve the method which has been given in the paper. Both journal, more repeats, better control of variables. That is, you can answer that you can improve by repeating the experiment or by controlling the different variables which has been used in this experiment or investigation. So improve measurement accuracy, better operators, you can use the better operators. So these can be the possible answer for that. So the variables and reliability of the data, all this can be included. All this can be used to answer your paper. So this part tests your ability to judge whether the experiment is good or flawed. So in this way, first of all, you have to, in these three components, I will summarize. That is, in the first, in planning, you must have to know how to design an experiment. And during the designing, you must know all different parts of the experiment. You must know what are the independent variables, what are the dependent variables, and what are the different operators uh, which you must use during the that investigation. And secondly, how to process the data, how to process that data, how to draw a graph, and how to know that the which part of the graph and which are the different what are the different patterns has been shown in that graph. And the thirdly, you have to evaluate the data, that whether it is working or not working, whether the experiment is good or flawed. That is according to this, according to the that data which has been given in the paper, you must know that is this is the this experiment is good or it has some flaws into it. So beside these components, there are some statistical tests which are part of this paper, which you must know. That the first test, which is some in some, so you can say any one of these tests can be the part of your paper 5. So you all must be familiar with all these statistical tests. So the first test which can be the part of the paper, that is the T test, student's test. So this test is used to compare two means. So when to use, you have two sets of quantitative data, two sets of quantitative data, then you will are able to compare of these. So want to know if the difference between the means is significant, then small, simple size actually should be less than 30. So what you conclude, what will be the part of this t test? That is the if calculated t value is greater than the critical t which has been given. So significance difference will be, it will be a significant difference. But if calculated value of t is smaller than the critical test, it means no significant difference. So keeping all this in mind, there is a formula for to find out the t-test. You don't need to learn these formulas because it has been written, it will be written in your paper. So it is you can see the this formula of the t-test, where t is the t value which you have to find out. So x bar is a sample mean, or mu is the population mean, and mu is the simple standard deviation, and n is a sample size. So these are the different, you can say, part of this formula of the t-test, and you have to find out during this solve when you are going to solve the P5. Next is chi-square test, which can be the part of the paper 5. The purpose of this chi-square test is to compare observed counts or expected counts. Sometimes we say that the observed value is different from the experimental values. So expected value is different from observed value. So on this, you can say categorical discrete data. That is, there is a difference in the data in the expected variable or in the observed reading. There is a difference. Then we use this formula, chi-square is equal to sigma and observed minus expected square divided by expected value. So where chi-square is test statistic, sigma is the summation sign. So all these are the statistical terms which you must know, don't need to learn, but it will be given in the paper. Sigma is the summation operation, O is the observed frequency, E is the expected frequency. So how can we interpret for this formula? The chi-square calculated is greater than the critical, then the significance difference is there. But if it is less than the critical, then there will be no significant difference. Then we move towards the third statistical test, which can be the part of P5, that is Spearman's rank correlation. So purpose of this Spearman's rank correlation is it is used to test for a correlation between two variables used when data is ordinal or continuous. Want to know if there is a relationship between the values, between the given values in the paper. The that is equal to 1 minus 6 sigma d square over n bracket n square minus 1, where d is the difference between the ranks, n is the number of observations. Then how can we interpret this? Plus 1, perfect, if the answer is plus 1. So it is perfect positive correlation. If it answer is minus 1, so the perfect negative correlation. If the answer is 0, it means there is no correlation between the given data. So compared to critical value. Then we come to the fourth test, which is the standard deviation. And the purpose of standard deviation shows spread or variability in data, comparing reusability of results, interpreting error bars. So if there is error. So this is the formula. You must familiar with these formulas. So these small standard deviation data close to mean. If there is small standard deviation, small deviation from the original value, then it means it is very close to the mean. So it is reliable. Large, it means data spread out, less reliable. Then we move to the last statistical test, which can be the part of the P5, which is standard error. So the purpose of shows accuracy of the mean. So this is the formula which has been given. So small standard error means is a precise value. Large standard means uncertain value. So in this way, these tests can be, any one of this can be the part of your paper, and you have to solve the paper according to all these values. That is, you must know all these things which I have described in this video. So to solve this paper, you must practice the past papers as well. You get familiar with the questions. So if you want to ask anything, you can message me in this video. You can message me, and then you can ask anything you want to ask. And please like, share, and subscribe my channel to watch more videos on paper 5 and 4 as well. So thanks for watching.